Hello, biologists. Today we're going to review systematics, species concept, and then we're going to talk about the Hardy-Weinberg equilibrium. So in systematics and taxonomy, taxonomy is the way that you name something, and systematics is the way that you figure out the relationships between something. This is what we did last term. We identified the different categories of life from the biggest domain down to the smallest, the species, and we practice using binomial nomenclature, which means the two-name system for each species. For example, that red fox right there, its Vulpes vulpes. Its genus is Vulpes. The genus gets capitalized for the V, and its specific epithet is also Vulpes. The specific epithet is not capitalized, and we always italicize the Latin name. When we refer to different species or genera or family in biology, we call them taxa. So taxa are different biological groups of organisms. And we have a lot. So this is a reminder of all the different species and organisms and phyla and classes that you learned last term, just to bring it back to the front of your head. You're not going to get retested on it, but we're going to talk about different animals a lot throughout the term, and it's nice to be able to organize them into our categories of what kind of critters they are and how they relate to each other. There's a couple things going on here. Some categories are split up into their different groups, and some are kind of lumped into one big group here, like calling this all vertebra. That's based on how you organize things in your head. And we generally say, as biologists, you're either a lumper or a splitter. So if you're a lumper, you like to take all these different subspecies and put them into bigger groups. You like to organize things in the big picture. Splitters like to break things apart by minutia. They like to think about the variances. They like to think about how each little thing is different. But it's person to person. I tend to be a lumper, but there's nothing wrong with being a splitter. And it's not like this is a real category. This just helps us think about how humans classify information in our brain. It's been a conversation for a while. So we have a comic here from 1969. And then we have this quote from Charles Darwin. Take a moment to read that quote. He is right. It's vague and arbitrary that we classify things as we do. Let's talk about how we classify them. We can classify them based on the biological species concept, in which if you breed and you have viable, fertile children, then you're the same species. I have two pictures of meadowlark on this slide for you. One is the eastern meadowlark, one is the western meadowlark. They look really similar, right? Like they look super similar, but they don't sing the same song. And because they don't sing the same song, they don't recognize each other as the same species, and they do not breed. So this is a good example of the biological species concept working. When it doesn't work is when we have something that reproduces asexually, or for extinct organisms, so we can't see them reproduce anymore, or for things that kind of blend over time, like ring species, or for fungi that have mysterious sex life, so we're not really sure what's going on there. If we were to use the morphological species concept, we would probably make those two meadowlarks the same species because they look so similar. The problem with the morphological species concept is it doesn't take into account convergent evolutions where distantly related organisms can look similar, and we'll show you an example of that in a second. And it doesn't take into account variation among individuals of the same species. So if I was looking at fossils of men and I found a fossil that was this big, and I found a fossil that was this big, using the morphological species concept, I might think they're different species, when really they're the same species and they're just different sizes. So let's talk about convergent evolution. Here, I have 3 mushrooms. Each of them looks really similar. Fungi are usually classified based on the morphological species concept, although that's changing. And so how do I tell these things apart? I'm showing you this not only because I love mushrooms, but spring is morel season. So if you're going to be going outside and hiking, keep your eyes open for these mushrooms. We have some really incredible edibles. Morchella elata is the real morel. This is the true morel. This is a wonderful edible. People will pay tons of money for it. It's highly sought after. It'll be at fancy restaurants and grocery stores all spring. And this mushroom has a single stipe. It's kind of cream colored, and it has a convoluted, pitted, contorted cap. So now let's go to the next mushroom. This is Gyromitra esculenta, and it has a similar structure. It's got a single cream-colored stipe. It's got a convoluted, pitted cap. It's kind of brown, but these can be brown too, so that color is variable. And some Gyromitra are black, like this morel in this picture. And then we come to our last one, Verpa bohemica. We have a single cream-colored stipe. We have a pitted, convoluted cap. This is also a false morel. So here we have our only real edible. These are questionably edible. The Gyromitra is considered toxic, even though some people eat it. The reason it's considered toxic is because it has a chemical in it that's really similar to jet fuel. And if you don't cook it the right way, it's going to make you sick. However, if you cook it the wrong way, that chemical will volatilize, and the gases of it will also make you sick. And so I always tell people, don't eat it, don't even bother. Some people do, but I would not recommend it. Then Verpa bohemica, it is considered edible, but not desirable. It's kind of got a slimy texture, and it does make some people yak, but it's not toxic. So it's not really edible, but it's not edible. It's just not particularly good. Some people like it. It's all up to you. This one, however, edible and wonderful. We know this one is edible. We consider this one very prized, but if it's not well cooked, it can make you really sick. So you do need to cook this appropriately. And even if you cook it appropriately, some people just kind of yap with morels. So as a heads up, try everything in a little bit first. So speaking of our morphological species concept, to get us back on track, how do we tell these apart when their characteristics are pretty similar? We can cut them open and look at the inside. That's a good hint. For a true morel, the stipe will always attach to the cap continuously. So it goes right up the base and continues on, and the stipe is hollow. Gyromitra, the stipe attaches to the cap, but it's all convoluted. It's not one central hollow cavity. It's like a mess of hallways. And then your Verpa, the stipe continues up, but look, here's the beginning of the cap. It passes the beginning of the cap and then it attaches up at the top. And so it's not continuous with the cap. And it's filled with this cottony layer. That's a dead giveaway that this is a false morel here. So we want these hollow chambers only, not convoluted or cottony. So I said that we use the morphological species concept for fungi, and that's true, we do. But to publish nowadays, you actually need the next species concept as well, which is the genetic species concept. The genetic species concept is great as long as you have DNA, as long as you can agree on how different the DNA needs to be, and you can agree on what type of DNA to use. So down here, we have the genetic markers that we usually use for animals. Several of these are from the mitochondria, from plants. Several of these genes are from the chloroplast. The ITS gene is from the ribosome. Bacteria have their own groups. And then fungi and protists, they are mostly using ITS, which is ribosomal, but they're also going to use their other specific DNA marker. These are also ribosomal back here. So they're spacer units for inside these subunits of the ribosome. We take the DNA, we compare it across all of our different specimens. We put the information into a computer program. The computer program figures out the differences between the DNA, and based on those differences, will produce dozens to hundreds of phylogenetic trees. It will continue producing phylogenetic trees until it finds the one that is most common. And the one that is most common, we call the most supported or the most parsimonious. In that tree, we're going to measure how well supported each branch is using numbers. So it's from 0 to 100, and that represents a percent, or it's from 0 to 1, and that represents the whole number of the percent. So here I have a phylogenetic tree comparing my true morels to my false morels, my verpas, and you can see the different locations where these taxa were collected. a lot up in Oregon and Washington, and then we start to get out of Oregon and Washington into Alaska, and down here we have Illinois, Michigan, Pennsylvania, Missouri, and we have our outgroups that we're looking at. We have China, we have Hungary. So we have a whole lot of different types of false morels and real morels. And we're comparing their genes to try and get these overall groupings to figure out how they're related. So here's another phylogenetic tree. In this one, we are comparing different viruses. And now our numbers are shown as 0 to 1. If you're bigger, you are more supported. So this 1.0 here, if I were to turn that into a percent, it'd be 100%. That means that all of the trees that were created by the computer showed that these two viruses are sister taxa. This branching is very well supported. Here as well, this sister taxa, the branching is very well supported. This one here, slightly less supported, and this branch, it's not quite sure about. So the lower the number, the less positive we are that that branch is supported. And by supported, we mean was the most likely option given the data we put into the computer program. Here's another visual. First, can you tell what kind of taxa are on this tree? Can you find out which branch is least supported? Where's the outgroup? And is this tree rooted? Go ahead and pause the recording and take a moment to answer those. All right, what taxa is on this tree? We've got a whole lot of plants. So here I've got Abies, that's fir, Cedrus is cedars, Pinus is pines, Pseudotsuga, our Pseudotsuga menziesii is the Doug fir, that is the state tree of Oregon. You know all about ginkgos and cycads from learning about them last term. We got our welwitschia, that weirdo that grew in the desert with only 2 leaves. So this is all plants. And then did you find your least supported number here? It's going to be the lowest number you can find. Who is the outgroup? Who is the least related to everybody else? This is going to be our outgroup right here. This is going to be liverworts. And then is the tree rooted? No, there is no root at the base of the tree. If there is a root, there'd be a little projection sticking off. That's a choice. It doesn't really matter, but it helps us figure out whether or not there is a projection into another relationship not being shown. All right, one more phylogenetic tree. Which is the outgroup, and are there 6 taxa? And then down here, one more question. Are house cats more closely related to cougars or bobcats? So pause the video, go ahead and answer the question. All right, who is the outgroup? We're going to go to the end of our tree. This tree is rooted. We've identified that these are all cat lineages in the Felidae. And then who is least related to all the others? That's the Panthera group. So our lions and tigers and our cloud leopards are least related to everybody else. Are there sister taxa? Yeah, these two are sister taxa right here. And then we have sister taxa right here with the cougar and the cheetah. That feels a little weird to think that those two are closely related. And we have another sister taxa right here with 2 other types of Felidae. And then the last question, who is most closely related to the house cat? Cougars or bobcats? So you're going to follow your tree backwards. Here's your domestic cat. Here's a branch. And then at our next node here is where we get cougars. So cougars are more closely related to domestic house cats than bobcats are because they're back another branch into the lynx lineages. So even though bobcats morphologically look a lot like house cats, like if you've ever seen a bobcat and a house cat together, you'd be like, okay, I see the similarities. And a cougar does not look at all like a house cat. Cougars are massive and terrifying. We can see on this phylogenetic tree that the morphological species concept wouldn't be a good way to set this up. This is based on genetic data instead. Let's get into the Hardy-Weinberg equilibrium. So Hardy and Weinberg were mathematicians, and they figured out that they could determine whether or not a population was evolving by setting up a statistical test to see if there was allele change or genotype change between generations. If there is no evolution, that means that we have to assume a couple of things. First, we know mutations lead to evolution. We know that gene flow, which is immigration and emigration, lead to evolution. We know that natural selection and sexual selection lead to evolution. And we know that a small population is affected by changes more drastically than a large population. So for there to be no evolution, you would need a huge population size, an infinitely large population size. For a species to be in Hardy-Weinberg equilibrium perfectly, none of those things must exist. So those are our Hardy-Weinberg equilibrium assumptions. Now we know that in real life, that's not really true, right? Mutations are going to occur. You're going to have some animals leave and some join your population. Your population's not going to be infinitely large. But this is a theoretical idea that these mathematicians use to come up with these equations. And now we can use these equations to compare real life to. So it's a statistical test of a theory, and theory meaning more of like a hypothesis of what it would look like if there was not evolution occurring. What are the equations they came up with? They came up with two. First, they came up with one to refer to allele frequency. In this equation, P is going to be the dominant allele, always, and Q is going to be the recessive allele. To figure out the allele frequency, it's P plus Q equals 1. So this is the first of the two equations. To figure out the genotype frequency, which is how many are homozygous dominant, how many are heterozygous, and how many are homozygous recessive, we're going to use P squared. That's going to be homozygous dominant. Q squared is homozygous recessive. And 2PQ is going to be your heterozygotes. Add those all together, and it equals 1. Let's take a moment to break this down. If I have an allele, and I have two options for it, I have a dominant allele and a recessive allele. All of those alleles together would be my full population, which is why it equals 1. For genotype frequency, if I know the frequency of each genotype in a population, it's going to equal the whole population. That's why it equals 1. So let's take a moment and let's work through a couple problems, and then the rest of this lecture, we're going to practice them. They are difficult, but you can do difficult things, and repetition really helps. So first, let's start with some pea plants. Pretend, as I have in my garden, you planted a bunch of pea plants. I'm really excited for my peas to get going. I love peas. And some of your peas are going to have yellow pod or yellow seeds, and some of your peas are going to have green seeds. So you are curious to know if you can figure out the allele frequencies. So here is your data. From all of the peas that you grew, you had a genotypic frequency of 0.49 that had the phenotype yellow. You know that this yellow was homozygous dominant. What does that frequency mean? That means that 49% of your population had this genotype. Of your other yellow pea seeds, you have your heterozygotes, meaning they have one of each allele. The genotypic frequency of this was 0.42. 42% of your seeds were this type of yellow. And then you have your homozygous recessive allele. You have 0.09%, or 0.09 being 9% of your population. Here, if we have 500 individuals, which means 500 of these seeds, and you know the frequency, you can determine how many seeds that is. If you know how many seeds that is, we can figure out P. Remember, P is going to equal the number of dominant alleles. To figure out the number of dominant alleles in the total population, which is the frequency, you're going to take all of the alleles that are dominant divided by all the alleles in the population. And that's a little bit wonky, so check it out. If I have homozygous dominant, those seeds have 2 dominant alleles. But I also have heterozygotes. Each of those seeds has 1 dominant allele. So to figure out how many times in my whole population the big Y shows up, I'm going to do each homozygous dominant individual times 2. So 245 plus 245. And then I'm also going to add 210 because every single one of my heterozygous seeds has one of the big allele. So my total is going to be 245 plus 245 plus 210 equals 700. And now I'm going to divide that total number of times that the big allele is represented in my population, divided by the total number of alleles in my population. I know that I have 500 individuals. Each individual is diploid. So I'm going to do 500 times 2. That's my total number of alleles in the gene pool. So I'm going to do 700 divided by 1,000. That equals 0.7. That's P. How do you feel? Okay, let's do the same thing for Q. Q is going to be the number of times the recessive allele shows up in your gene pool. Your gene pool is your total number of individuals times 2. You times it by 2 because each individual has 2 alleles. So here, each of these individuals has 2 recessives, and this individual has 1 recessive. So I'm going to take 45 plus 45 plus 210. That's the number of times this little y shows up in the population, divided by the total of the alleles in the population, and that equals 0.3, which is Q. So I can always double check my work. I know that P plus Q should equal 1. Did the answers I get equal 1? Yes, they do. All right, I'm feeling good. So now that I know my allele frequency, I can determine my genotype frequency. So I'm going to click to the next one. Once you get P and Q, we can use that to plug into this equation, and we can figure out our genotype frequencies. P squared is the number of dominant homozygotes. Q squared is the number of recessive homozygotes. And 2PQ is the heterozygotes. So why is this number set up like this? It's all related to the probability of what you would get with these alleles based on a Punnett square. So if I were to set up a Punnett square comparing my 2 alleles, I would find that the chances of getting a homozygous dominant P is P squared, and the chances of getting a homozygous recessive P is going to be Q squared. The chance of getting either PQ or PQ, because there's 2 of them, is going to be 2PQ. So I know P equals 0.7. So to figure out my number of homozygous dominants, I'm going to take 0.7 squared equals 0.49. And that is how much of my population is going to be homozygous dominant. Same thing for Q. I like doing the squares first. So Q squared, 0.3 squared equals 0.09. 0.09 is the predicted frequency of homozygous recessive P's in your population. Then I have my heterozygotes. My heterozygotes are going to be P times Q times 2, which is 0.42. And that is the probability and the frequency of getting a heterozygote in my P population. So I know that was a lot. Let's practice. Here, I've got a beautiful picture of my Parker boy. And I want you to pretend that you found a population of cats who only have black fur or white fur. The black allele, big B, has complete dominance over the white allele. If there are 1,000 cats, 360 have the homozygous dominant genotype, 480 are heterozygous, and 160 are homozygous recessive. What are P and Q? Go ahead and pause the recording. Please give this a try. All right, so did you work it out? We have 1,000 cats, so I know my total population. I know my amount of individuals with each genotype. That means I can figure out the frequency of my alleles. To figure out the frequency of my dominant alleles, I'm going to take the number of homozygous dominant plus the number of homozygous dominant. Why am I counting it twice? Because each of these cats has 2 of these alleles. So 360 plus 360 to account for each of these big Bs, plus the number of heterozygotes, because each heterozygote has a big B. So 360 plus 360 plus 480 divided by the total number of alleles, not the number of individuals, the number of alleles. Because cats are diploid, each cat has 2 alleles. So it's going to be 1,000 plus 1,000. So I'm going to take this number, divide it by 2,000, and I'm going to get 0.06. I'll do the same thing for the recessive allele. 160 plus 160 to account for both of them, plus 480 to account for that little b, divided by the total number of alleles, which is my population size times 2, gives me 0.04. I can double check my work. Add those together. Do they equal 1? Yes, they do. Now let's figure out the genotype frequencies using P and Q. So P was 0.6, Q was 0.4. Do you see that Max is in this picture too? Here's his little ear. He's watching Parker. Go ahead and pause the video and get your answer. Here are our answers. We took P, squared it, got 0.36, took Q, squared it, got 0.16, and then figured out P times Q times 2 equals 0.48. What does that mean? That means if I look at this populations of cats, about 36% of them should be homozygous dominant, 16% of them should be homozygous recessive, and 48% of them should be heterozygous. Let's practice another one. You revisit the same cat population 1 year later because you want to compare the new generation to the old generation to figure out if this population is evolving. Remember, evolution occurs over time. It doesn't happen in individuals. It happens in a population's genes. So you have to compare generation to generation to see if a population is evolving. In this next generation of our black and white cats, we have a total of 800 cats. 672 are black, 128 are white. Use this to figure out both the genotype frequency and the allele frequency. Pause the video, please try it. All right, here's how we did it. I have 800 cats. I know that the black ones are both going to be heterozygous and homozygous. So I can't use that yet, but I can use the homozygous recessive. So if I know that these homozygous recessive are 128 of the 800, I can figure out what Q squared is. So Q squared is the number of homozygous recessive divided by the population size, and that's Q squared. So my Q squared is 128 divided by 800, which gives me 0.16. I square root that to get Q. Once I have Q, I can figure out P. Once I have P, I can figure out the genotypes of P squared, Q squared, and 2PQ. So ask yourself, now that you've done this, has evolution occurred in this cat population? No. Evolution has not occurred in the cat population because the genotype and the allele frequencies are the same. So they are not having microevolution. There's not some force changing their genetic makeup from generation to generation. If there is not evolution occurring, we say that this cat population is in Hardy-Weinberg equilibrium. So saying that yes, it's in equilibrium means no, there's no evolution occurring. And it will ask you both ways. So you're going to have to say yes and no to mean the same thing. So yes, Hardy-Weinberg equilibrium means no evolution. All right, let's try another one. You find another population of cats, but this time the cats are mostly black, mostly gray, or mostly white. So we have different genotypes, different phenotypes. There are 210 black cats, 245 gray cats, and 45 white cats. Go ahead and determine the genotype and the allele frequencies. And please look at how helpful Max was being while I was trying to frame a poster. So pause it, go ahead and try. All right, so here's our answer. We have 210 of the big alleles for our homozygous dominant individuals, 245 of our heterozygotes, which means we can do this in two ways. Either we can do it the first way we did, which is you take the number of homozygous dominant plus the number of homozygous dominant plus the number of heterozygotes. So this is all of the dominant alleles in this gene pool divided by all the alleles in this gene pool, which is your total population size times 2 to give us P. Or you could do it the way that we just did. We know that P squared is going to be our number of homozygous recessive individuals divided by the total population. That is the frequency of the population that has the genotype homozygous recessive. If we do 45 divided by 500, we end up with our frequency of that genotype, which is 0.09. And then, now that we know Q squared, we could square root it and figure out what P is. So P will be 1 minus the square root of Q squared, which is 0.7. So we got the same answer in two different ways. Once you get P or Q, you can figure out the other because P plus Q equals 1. And once you have both P and Q, you can figure out the genotype frequencies, which is P squared equals 0.49. So 49% of the population had black fur. 2PQ is 0.42, which means 42% of your population had mostly gray fur, like this handsome kitty here. Or you had Q squared, which is 9% of the population had white fur. Here's another practice. Same cat population with black, gray, and white fur, but you come back the next generation, you find that the population size is 400, and there are 64 mostly white cats. Go ahead and do the math, and then answer the question, is this population in Hardy-Weinberg equilibrium? You're going to take your frequency of homozygous recessive genotypes, which is the number of homozygous recessive you have divided by the population size. We had 64 mostly white cats and 400 total cats, which means that Q squared equals 0.16. Now that I know Q squared, I know Q, and I know P. P is going to be 1 minus the square root of Q squared equal 0.4. And now that I have P and Q, I can figure out my genotype frequencies. Note, genotype frequencies are how often these sets of genes come together in individual cats. Allele frequencies are how many times the alleles are in the whole population, regardless of how many cats there are. So is this population in Hardy-Weinberg equilibrium? No, it's not, because P and Q changed and my genotype frequencies changed, which means there has been evolution in the gene pool. And so we're not in Hardy-Weinberg equilibrium, which means this population is evolving. All right, one more go with a different organism. Here I have a green anole lizard. Coloration of lizards has complete dominance, where green scales are dominant over blue scales. There are 1,024 individuals with the genotype dominant, 512 individuals that are heterozygous, and 64 individuals that are recessive. Use this information to determine the allele and genotype frequencies. Go ahead and pause it and give it a go. Here are our P, Q, and our genotype frequencies. Now you know you can't just do it once, you have to compare generations. So we're going to continue. The next generation of the same anole lizards has 1,092 individuals with green scales, green being dominant, and 108 individuals with blue scales, blue being recessive. Go ahead and use that information to solve for P and Q, and then answer, is this population in Hardy-Weinberg equilibrium? So here we are, P equals 0.7, Q equals 0.3. Are we in Hardy-Weinberg equilibrium? No, because our allele and genotype frequencies have changed. There is a force acting on this population that's creating microevolution, and thus this population is evolving, meaning it's not in Hardy-Weinberg equilibrium. All right, I hope that was fun. Math problems are always really fun. Anytime you need practice, feel free to Google practice questions of Hardy-Weinberg equilibrium. There's a lot online. All biology classes are going to teach this type of population genetics, and so you can find a lot of resources online. And email me with any questions. I would love to talk about it. I hope you have a great day, and we'll chat soon. Bye.